I'm Annelie Jönsson, and I work at the Department of Biology at Lund University. I work with a data host project, just like Lisa was telling you about, and which was assigned to us by the Swedish Environmental Protection Agency. So we deal with data sets from systematic monitoring on birds and butterflies mainly. I will, in this presentation, I will tell you which data are available, or at least will be available from us, a bit about how they are produced, and then I will focus on how you can find them on SBDI, and I will also mention a few things about what it is you're actually getting when you're downloading data. And I will give this presentation to you as potential data users as well as publishers of data. That's my angle. So here's the full list of data sets that we are responsible for in our data host project. On the left here, we have two major schemes, the Swedish Bird Survey and Swedish Butterfly Monitoring Scheme. They're actually managed by my colleagues in-house, so to speak. Both schemes are financed by the Swedish Environmental Protection Agency, and all 21 county administrative boards they collaborate with and support these schemes. And the mission is, of course, to monitor population and distribution changes among Swedish birds and butterflies, and a bit more recently, also mammals. On the right, instead, we have some external data sets from survey programs that are managed mainly by the county administrative boards, länsstyrelserna. And I will use this in-house monitoring scheme to describe, as an example, to describe the kind of data that we have and why they are actually systematic. Let's point out we have the oldest data set we have is the waterfowl count that actually starts in 1967, and the most recent program we have is the Swedish coastal bird monitoring that started in 2015. Anyhow, the fixed routes from the Swedish bird survey, the so-called standardrouterna. I actually chose this background picture to illustrate that sites are systematically placed without regard for the convenience of the surveyor. The view that you're seeing is the actual path facing a surveyor who's about to count birds along a transect on an actual site. In the program, there are 716 squares, and they are evenly distributed across Sweden. So this means that each habitat in Sweden is surveyed in proportion to how much there is of it in the country. Of course, all the sites are surveyed using a standardized method, a systematic method, which is, like Marcia said, repeated year after year after year. The sites are in this survey 2 by 2 kilometer squares. They are visited once a year in late May to early July. Each site consists of 8 1 kilometer transects and 8 point counts. Along all these transects and on all these points, all birds heard or seen are noted, regardless of how far away they are. But in general, they're more or less plus minus 200 meters from the transect or the point. Some trivia from last year to show the scale of this scheme. We had 235 people walking about 4,400 kilometers while they observed 166,000 birds of 224 species. Almost all the species that we have in the database were observed. And data in this program stretches back from 1996, and the mammals were started in 2011. The data that is collected can obviously be used to calculate trends and distributions for these various species. The data can also be used for so-called miljömålsindikatorer. Not sure about the English word for that, but also for reporting to various EU directives, etc. But it's not only environmental monitoring we can use this data for, also basic and applied science, like also Matthias mentioned, the effects of climate change. So here we have a few examples of studies that have been done on these data. So basically the same data or different parts of these data can be used and reused to answer different questions. Here we have the full list of our data sets again, and the ones highlighted with yellow, they are the ones that we have published so far. So 3 of the data sets from the Swedish bird survey. We have published the Swedish butterfly monitoring scheme, and then 1 of the external data sets, the coastal birds in Gulf of Bothnia, kustfåglar i Bottniska viken. I will now spend the rest of the presentation to show you how you actually find these data on the SBDI webpage. So the first step is obviously to go to that webpage, biodiversitydata.se. On this page, you go to the Data and Tools tab. You click that, and you end up on this page. Here, under Swedish biodiversity data, you click on Datasets. That takes you to a page where all the datasets that are available on the SBDI are listed in alphabetical order. So you either scroll through or click through this list of datasets until you find what you're looking for, or you type in the whole name or part of the name here in this box and click search. As you can see, here we have the 4 datasets in our collection that start with the letter S. We have given the full English name of the program, but also included the Swedish name in the title, so you can search on both. Makes it easier to find them. In case you don't actually know the name of the dataset you're looking for, but if you know who's providing it or publishing it, you can also find and get access to the datasets through this link. If you go back to this page and click Swedish data providers, then you get a list of data providers in alphabetical order, and under each will be all the data sets that they're responsible for. However, I stick to this data set route when I continue, and I will explain why. So back to this list of our data sets, and we will look at one in particular to show you what it looks like. I've chosen standardrutorna again. So if you click on that name, the survey program's name, this is the page that you get to. Here's the top of the page, and here's the bottom of the page. I had to split it so I could fit it on the screen. But basically, you get the description that Lisa was talking about. You get some information about where the data is collected and why, something about the quality and methods, more about that later. And we have these lovely graphs that Lisa also explained to you. So this is the metadata that is provided. If you want to look at the actual data, you have 2 options. You have the button here at the top or the link further down the bottom. So if you click one of those, you get a long list of all the records that are in the data set. So of course we dive deeper and look at one record. I've chosen this one just randomly. This is what you see then. So this is all the data for this record that is in the data set, divided into sections for easier overview. So you have information about when it was actually recorded. You have information about the taxonomy, species, etc. And also, of course, where. There is a bit of information about data quality. Nothing major in this instance, luckily. There is just a warning to say that some data that is provided when you download the data has been sort of inferred or assumed. We didn't supply we didn't actually state in the data set that this species was present, but obviously because we have a number of individuals observed, it has been assumed to be present, which is entirely correct. Once you looked at this and you can see that the data is kind of what you're looking for and it will be very useful for you in your analysis, then you go back to your the previous page and you basically hit the download button and the data is yours. But what is it then you're getting? What are you getting when you're downloading data? I will show you that, and I will also mention a few things to be aware of. Like Lisa told you and showed you, we also publish our data onto GBIF through the IPT as Darwin Core archives. So they consist of 3 different tables, CSV files, the event, the occurrence, and the EMOF, the extended measurements or facts files. And all these 3 files are connected or linked together through unique event and occurrence IDs. When you download data from SBDI, instead you get one Excel file, which is great in one way because you don't have to fiddle about with linking tables together with IDs. Unfortunately, the data that is in the EMOF will not be included. This is because GBIF doesn't yet support that extension and they can't actually include it in the data that is published. Now, I was going to show you, well, Lisa already showed you what a Darwin Core archive looks like. So we have the 3 different tables, and this is supposed to be the filter, just to show you what kind of information could be in the EMOF, so information that you could be missing out on. This example is a survey program where it's, in some counties, it's mandatory to count either pulley, and in some places it's voluntary. So here we have included the information about whether the surveyor has counted them or not, so that the data user knows whether to expect this data or not. Likewise, the pulley, when they are observed, they should be ranked according to size. So that data is also put in the EMOF because it doesn't fit in the other tables. There is no Darwin Core term to that equals pulley size class. But there is a way around this. There is a way of getting this data, and that's if you actually go to GBIF. I know we're doing an SBDI webinar, so we shouldn't talk too much about GBIF, but I think it's important to know where you can get all the data. So if you pop over to GBIF quickly, then you go to a datasets page, you click the download button, and then you choose this alternative, the source archive. Then you get what Lisa showed you, the 5 different files, which includes the EMOF. You then have to link them all together yourself, but at least you got everything exactly how we publish it onto GBIF. Some other things that I've noticed when I've been comparing what's been published with what we actually uploaded. There are some fields that are missing from the event and occurrence tables. I don't know why yet, but I've used the support form to highlight this, and hopefully we'll get an answer. Also, both GBIF and SBDI, they run your data through some validation process in this publishing process. And for certain fields, there are specific code lists or vocabularies that are used. So if your values that you have used in this field, if they are not actually on that vocabulary, then you might end up with an empty cell. And but SBDI actually provides a table that gives you a comparison of the original versus the process data. So when you're looking at one of those specific records, this is the same view that I showed before. So you've chosen one of the records in a data set. This is what you see. There is this button here, the compare original versus processed values. If you click that, you get this table where you have a column here with original values, as in how we actually supply it when we upload it through the IPT, and then a column to state what has been filled in the validation process. In this example, there was nothing major wrong or changed. The only thing I could spot was really that we supply scientific name as just Embellida hortulana, and the process has filled in the author and the year and the scientific name. But otherwise, this was fine. And lastly, just a couple of words about metadata. When you download the data from SBDI or the recommended route on GBIF, you don't actually get any metadata in the download. If you choose a source archive, you do. The page I showed you earlier as well, the metadata on the web page, we go back to that as well. This metadata page here with a description and everything. At the moment, at least for our data sets, there is some missing metadata, like the method section. That is not properly filled in here. We have supplied a lot more information about methods, but it's, I guess there's a mapping problem or something for getting that information here. Luckily, there is a link to a GBIF metadata webpage. So if you click that link, then you will see all the metadata that has been supplied for this data set. And that was all I had for you. On that note, I say thank you very much. And if you want any more details, please ask or contact us.